the components بتوع theoretical و practical part. ثالث جزء how marks are calculated. ورابع جزء ال first chapter في ال AS. هشرح لكم دلوقتي نظام السيشن. إن شاء الله في أول السيشن كل الناس هيتعمل لها unmute، وبعد كده أول ما بتبتدي مس دكتورة إن هي تشرح بيتعمل mute all. في حال إن في حد عنده أي سؤال بيكتب على الشات، وميس هدى كل فترة هتقف وهتاخد الأسئلة وهتجاوبها إن شاء الله. ميس هدى حضرتك اتفضلي.
I'll repeat again. Seems like I didn't unmute myself from the very beginning. I'm sorry. Okay. Good evening again. Thanks for the introduction. Today I'm going to talk about the whole A-level in the two stages, AS and A2. Okay. And I'm going to start with the AS topic today. And I will tell you what you gonna do, what you need, okay, to get not only a high grade in AS, which is the highest is A, but also how to achieve high marks to take it with you, okay, so that you can add it to the A2 marks and achieve the star, okay. I need to share the screen now, okay. Then I'm going to stop the video. The A-level biology have two major procedures. It's either you take it as linear, and this means that you are going to take the AS plus the A2 in same exam session. Or you can take it modular, and this means that you gotta sit for AS only, and then you sit for A2 in a separate session. Sorry, A2.
Mrs. هدى بعد إذن حضرتك ممكن تعملي share screen?
I didn't share it. I shared screen.
No, it didn't work.
Share screen. I'm sorry. Now it's shared.
Perfect. تمام. Thank you.
Okay, I'm sorry. I'll repeat again. Your A-level, your A-level biology, okay, you can take it as linear. This means that you are going to sit for AS and A2 both in same exam session. Okay, or you take it modular. This means that you are going to sit for AS first, and then after that you sit for the A2 biology in a separate session. Okay, let's see together what are the papers we are going to sit for and what are the skills for each one. As you see here, that our syllabus code is 9700. This is the syllabus code for the Cambridge syllabus. Okay, 9700. Okay, and the papers you are going to use for this are number 1, paper 1. Okay, and this is the first paper. Okay, paper 1, it's a multiple choice paper. You have 40 multiple choice questions. Okay, and then these are awarded for 40 marks. Okay, and it makes 31% of the AS mark. Okay, another paper, which is paper 2. Okay, and this is a structured questions. To enter the AS level, you must pass or you achieve a high mark in IGCSE. If you have sit before to IGCSE, so you already done paper 4 in O level. Paper 2 is like paper 4. Paper 2, okay, is a structured questions. It has 60 marks and it makes 46% of the AS mark. Paper 3, as you see, it is titled Advanced Practical Skills. You have 2 or 3 questions, okay? Not necessary to be all experiments. You may have some experiment or some activity done using a microscope. We will go through this separately now. The exam takes about 2 hours and it's awarded for 40 marks, and this paper makes 23% of the AS mark. When you finish your AS course in a session and you need to complete it, then you will start to sit for paper 4. Okay, it's a 2-hour and it has 100 marks and it makes 38.5% of the A-level mark. And there's another paper, which is paper 5. It's not practical, but it's based on practical concepts which you have studied already in paper 3, okay? This is why they title it Planning, Analysis, and Evaluation. And this paper is awarded for 30 marks, and it is 11.5. Suppose that you are going to sit for the whole A-level in one session. This I mean the linear A-level. You are going to sit for all the papers in the same session. In this way, the weight for paper 1 will differ from that of the AS, okay? As you see, paper 1 and paper 2, and paper 3 also has different percentage from the whole A-level. So again, students willing to take A-level biology have the choice to do it modular so that they can do the AS in one session, and then they do the A-level in another session, or they may choose to take the linear A-level, all of it, okay, in one session. In this case, he will sit for 5 papers, okay? This is what you need to know before starting, okay? I need to talk a little bit about the practical. This is one of the, I cannot consider it as nightmare, but some students taking the AS consider that the hassle in AS is the practical. Actually, it's not. As long as you get the skills, it's no more a hassle for you, because it's not about taking doing the experiment and giving results. No, it is also about knowing how to handle the question, considering the analysis of what are the sources of errors and how could you avoid it. I have some problem. Okay, practical is also related to the topics you study in AS level. In AS level, you have 11 topics: cell structure, biological molecules, enzymes, cell membrane and transport, mitotic cell cycle, nucleic acid and protein synthesis, transport in plant, transport in mammals, gas exchange and smoking, infectious disease, and immunity. I know that all these topics, if you have done your IGCSE level, are not new for you. You have studied them, but on the O-level standard. Here in AS, the issue is different. You have more deep information. You have more details, okay? And you may ask, what about cell structure? What I'm going to do in practical for it? Okay, as you cannot see cells by your naked eye, so you can know that you are going to use a microscope. Here comes the question, which requires the use of a microscope. In biological molecules, there is experimental work you have done already before the IGCSE, and you know about the food test and so on. Okay, so you're going to do them by yourself, and I will show you how to do them. Also, you will get some ideas about the enzymes, how they work, and how could you investigate the enzyme activity. Okay, let me go through this topic one by one. This is the first topic, cell structure. Okay, as you see, you are familiar with some parts in it. Like structure of a typical plant and animal cell, okay? And this first part has a practical application. Also, you're familiar with calculating magnifications, okay? So you will use, we will see drawings from photomicrographs and electromicrographs, and I will teach you how to do this. This is for you is totally new. Eyepiece graticule, using an eyepiece graticule. Okay, what's an eyepiece graticule? This is, we call it a ruler fixed on the eyepiece of a microscope. Okay, and then you calibrate it using something called stage micrometer scale, and you should be familiar with, and I think you are familiar with it, millimeters, micrometers, and nanometers. These are we use in cell studies, and I think you have an idea about how to measure millimeters and how to convert them to micrometer. I think the use of this unit is new for you. Again, you will know what is the difference between the term resolution and magnification, okay? And then you have to know a skill of how to calculate the actual size of a cell or part of a cell or an organelle or so on. Okay? What else you should know in cell structure? Like describe and interpret electromicrographs, drawings of typical animal cells. Those organelles, you need to know everything about it. Like the cell surface membrane structure and the size of it or the thickness of it, detailed structure of a nucleus, nuclear envelope, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi body, mitochondria, ribosomes, lysosomes, centrioles and microtubules, chloroplasts, cell wall, plasmodesmata, large permanent central vacuole. Okay, I know I'm too fast in this because I'm just going through. I will talk about in detail on each and every part of this. Also, we need to state that ATP is produced in mitochondria and chloroplasts, outline key structural features of a typical prokaryotic cell, meaning the bacteria, the size of it, and what it's made of, and detailed structure in it. And then we are going to compare and contrast between a prokaryotic cell and a typical eukaryotic cell. Okay. At the end, we will end by mentioning some key features about the structure of a virus. It's a non-cellular structure. Okay. We will study viruses in our syllabus, referring to its structure in this chapter, but also furtherly, we are going to go to another chapter called Infectious Disease. We'll talk about viruses. The next chapter is biological molecule. Again, the first part of it is concerned about the practical. Here you see part A, carry out tests for reducing sugar, starch, non-reducing sugar, and the emotional test for a protein. Okay, and then you carry out a semi-quantitative Benedict's test. I'll give you some hints, some very useful hints. In spite, we do not do practicals online, but I will give you a lot of hints concerning this, which are very important to help you do these experiments. And then we are going to start to talk about detailed structure of biological molecules like alpha glucose and beta glucose. And we'll illustrate the term monomer, polymer, macromolecules, giving examples of monomers such as the alpha glucose and beta glucose and fructose and so on, amino acids. Also, we're going to study some macromolecules like the lipids, starch, cellulose, proteins, and we are going to study some examples of proteins. In lipids, we're going to study or know about what are triglycerides and phospholipids. Okay? We will continue by studying proteins and water. Okay, you tell me what's about water. Water is H2O. What else we need to know about? No, water has a lot of properties. Okay, and those properties have a role in our systems, like maintaining your body temperature, like it's a solvent, like it is a medium for metabolic reactions and so on. Okay, next we will start the next chapter will be enzymes. You are familiar with enzymes, but we need to know more details about it, like how to investigate enzymes, progress of enzyme catalyzed reactions, effect of pH, effect of temperature, effect of enzyme concentration, substrate concentration, and what are inhibitors, okay? And we will go deeper in the enzymes for part you're not familiar with it, you didn't study it before, something called the Vmax and Km. This is what we call maximum velocity and the affinity of enzyme to a substrate. Next chapter is cell membrane and transport. We will mention cell membrane in more details, like not only the phospholipids, bilayer and protein. Now we will know more structures within cell membrane and its role in transport of molecules through it. Or if they are large enough, we will, study some new mechanisms you're not expected to be familiar with it before, like exocytosis and endocytosis. Endocytosis and exocytosis. This is when the subject is too large to move through the membrane. Next chapter is the mitotic cell cycle. You studied an outline about the mitosis, in your IGCSE, but here we will know more details about mitosis, and we'll extend our study then by studying the nucleic acid and protein synthesis. You have studied before an outline of protein synthesis, but here in AS level, we will go through a lot of details in the DNA structure, replication of DNA, and also details in protein synthesis. Transport implant, this is not a new topic for you, but we will go through detailed structures of xylem, phloem, a transverse section in a stem, transverse section in a root, how water, how assimilates move through the plants. And then we will study many transport mechanisms in how water move in xylem. Some pathways you are not familiar with it, like something called symplastic and apoplastic pathway. After we finish this chapter, we will go to transport in mammals. We will study the blood vessels, components of blood, and then we will introduce a new transport fluid called the lymph. Also details about tissue fluid and the differences between blood, lymph, and tissue fluid, as well as the structure of the heart and the details of cardiac cycle. Diseases are not part of our syllabus. But here in gas exchange and smoking, you will study details of gas exchange system, microscopic study also. We will know about details of the walls of a trachea and walls of alveoli and lung and bronchioles. And then we will study smoking and how this affects the function of the gas exchange system, and also its short-term effect of nicotine, carbon monoxide, and the cardiovascular system in the end. The next chapter is infectious disease. You studied last year something about cholera, AIDS, and malaria. Also, we studied them this year. We will know the pathogens, how the structure of it, how the mode of transmission happens, prevention, as well as the treatment of those diseases. Not only those, we will also study TB, we'll study measles, and smallpox. At the end of this chapter, we will know about the antibiotics, how do they work and mode of their action, and when they are used with each and every disease. Next chapter, and the last chapter, immunity. In immunity, in the IGCSE, you have studied something like called a mechanical barrier, a chemical barrier, and then the last line of defense, which was the action of white blood cells. In AS, we will focus on the action of white blood cells. We will talk about the origin of white blood cells, the difference between a phagocyte and the lymphocyte, and what is the primary immune response and what is the secondary immune response. And then we will study a very important hint in the end about what is a vaccination, what's made of, a vaccine is made of, and a new mode of treatment. It is totally new, which is the antibodies and the use of monoclonal antibodies. This is totally new. This is the coming up medicine. This is how the progress in medicine will, like diagnosing the disease, helps to diagnose a disease, because a lot of diseases have common symptoms. And in the very big, in the past, because of those common symptoms, sometimes treatment doesn't go well. So the use of monoclonal antibodies helped us a lot, okay, to discriminate if this is a symptom is due to this disease or not. Not only this, they use monoclonal antibodies to treat some diseases like cancer. They are, okay, although it's nowadays expensive, but researches are going on so that can help cancerous people, okay, to overcome this, and this is what we call a targeted treatment, okay. This is the end of my introduction. I will go again through some details about assessment during the A-level study. I will return back again to the components of yes.
Mrs. Hoda,
Yes.
there is one question. Someone is asking about the practical part.
Yeah.
Is it possible to master the practical part while being homeschooled, or is it tough to do?
Okay, dear. This is what I'm going to talk about now, okay? And this will solve your question. As I told you before, you will sit for paper 1, and this is an MCQ. And you sit for paper 2, and this is structured questions. And as I said before, these are the theoretical. Here, dear, what I'm going to talk more details about paper 3, which is the practical. In practical, as I said before, you have 2 questions. Question number 1, okay, which is about doing an experiment. If I am to break down question 1, okay, you will go, when you go through a question 1 exam, okay, in a practical, you will find out only 4 marks depend on experimental work. This is the part when you do the experiment and you get the results. I'll show you now a practical exam, okay? And the other 16 marks needs from a student to learn how to analyze, make conclusions, and use this to explain the biological phenomenon, to explain why this happened. Okay, again, question 1, which is the experimental work, only 4 marks of it depend on the experimental work and results. This is the part you do yourself, and you do the experiment and you do the practical procedures, and then you get your results. The other parts of the question depend on you. If you learned how to analyze, how to make conclusion, and how to use both these to explain why this happened during the experiment. The question 2 in the practical exam depends on microscope work. You will be provided by a slide, and you need to use the microscope to draw plan diagrams, which I'm going to teach you here how to draw plan diagrams. Also, detailed diagrams. Also, you will know how to take measurements, and I will also teach you how to take measurements from microscope. Okay, so practical for homeschooler, you can even, I will teach you how to predict results. And I will open with you now a practical exam to show you how it goes. Practical, as I told you before, makes 23% of your mark. Its raw mark is 40. But after your exam paper is marked, okay, and suppose you are a good student and you have got and you have got 36 out of 40. Okay? This is not the mark which will be added to your total. It will be adjusted. If you get 36 out of 40, it will be adjusted so that we know how much you get out of 30, because the AS total becomes 130. 130, 40 marks in paper 1, 60 marks in paper 2, and then 30 marks in practical. This is what makes the 130. Why? Because the A2 total is also 130, 100 for paper 4 and 30 for paper 5, because AS and A2 has a total of 260. Okay? Let's open a practical exam together now. I chose you now a practical exam, which was November 2012. It was variant 3. By the way, practicals has 5 variants. In the Middle East, in June, we sit either for variant 1 or variant 2. In November, we sit either for variant 3 or variant 4. Let's look together this practical exam. As you see, we have 2 questions, question number 1 and number 2. In the very beginning, there is advice from the examiner. You are reminded to have only 1 hour for each question in the practical exam, okay? And in this exam, there was a hypothesis to be tested, okay? Which is if the seeds, there was seeds provided for the students, and we need to investigate if these seeds release the enzyme which hydrolyzes the sucrose, introducing sugar, okay? You are provided with, these are the material are provided on the bench. And then here are the procedures the students should do. I'm scrolling because I'm going to do this with you later, okay? But here, there are some dilutions need to be made, and I will teach you how to make those dilutions. This is a skill you need to know before entering a practical exam. And I will explain for you 2 methods of dilutions. Something called simple dilution, and another method is called serial dilution. This I'm going to teach you how to do this. Okay, you need to understand how to use this method and how to use this method. Okay, and this does not require a practical work. It requires from you to understand the method and know when to use this and when to use that. After the student decides how much volume of Benedict you are going to use, because you know sucrose, when it's broken down, gives fructose and glucose. In this space, you are going to record your results. And I will teach you how to make a good record, an excellent record, because as you see here, this part is awarded for 4 marks. Guess what? One of those marks, if I need to break down those marks, 4 marks for what? The first mark is to draw a table with closed cells, like that. If you draw a table in this way, and then you record this variable here, variable then, and leave the table in this way, you lose a mark. This was what's called open table. I will return to this later on when we make a lesson concerning the practical part. One other mark is for the units of variables you are recording. Another mark for inserting data in table. And the 4th mark for the trend of your results. So look again with me. If you did not do the experiment, and you draw the table with closed cells, and you have inserted the variable in the right place with units, and you just inserted the data for the dependent variable because this is the thing you know before starting the experiment, and you left this part empty, you know what? You get 3 out of 4. And if you go through the rest of the question, you will find out something weird. The next part does not require any practical work. This is, you are now investigating or telling if the data support the hypothesis or not, and then you are going to explain using biological phenomena, and then here you are going to modify the procedure and suggest a further procedure. And at this part, you're going to draw a graph and get data from the graph and describe the trend. The graph is not based on your data, it is based on those data provided here. Okay, so question number 1, which contains the experimental part, okay, sometimes you can even guess the results, and I will teach you how to do this, okay? Because the most important thing here is to manage the other skills concerning explaining, modifying, and if the hypothesis is right or wrong, and how you estimate, and how do you make a table, and how do you decide to use what volume of Benedict, and so on. If you are not capable of knowing how to decide to do those, okay, so sitting in the lab or standing in the lab would do nothing, okay? Let's go for question 2. In this, I will show you here, I have done myself, taking a photomicrograph from a microscope so that I can explain for you the parts of the any section, a section in a root, in a stem. So I will show you some sections here in the lesson as if we are looking at the microscope together, and I will teach you how to make a plan diagram so that you can get those beautiful 5 marks. And then I will teach you how to make detailed drawing of some cells, plant cells, and types of them. And here's the part concerning measurements from microscope. Guess what? In this exam, the students were not doing it from microscope. They're doing here on the question paper, as you see. Here is the part which concerns the graticule I told you about. This ruler, this is what called eyepiece graticule. This is what fixed on the eyepiece, and this ruler is called stage micrometer. And I will teach you how to calibrate this eyepiece graticule using the stage micrometer, and then how to answer such questions, and then use it to measure the diameter of a xylem vessel as you see here. After that, okay, I will teach you how to make a comparison of this section with what you see under the light microscope. Here is the practical. I hope this answers your question. Okay? Let me now start with the first chapter. Before I start, I need to take some questions, Susie, if there is any. Is there any
We
question?
don't have any questions currently, Mrs. Hoda. Thank you.
Nothing? Is something not
All is free.
clear? I will give you some moments if you have questions concerning the AS.
In the beginning, someone was asking about the difference between A and A-level and AS, but you have already figured that out during the explanation. If you have a minute or so and you can explain it again, this would be great.
Okay, I will explain it again. Once again, what is the difference between A-level? A-level is the whole thing. Is the screen clear for everyone? I need to make the marker a little bit thicker. A-level biology. This is the whole thing. And it has a total of 260 marks. 130 are for the AS biology. Sometimes they refer to it as A1. And another 130 marks for the A2 biology. A candidate can choose two ways to finish his A-level biology. Either to take it linear, in this case, in one session, you sit for AS and A2. In this case, he will sit for 5 papers. Another candidate will choose to sit for AS first. Like, for example, if you choose to sit it in this November session, and then in another session, you sit for A2, okay? For example, for this November session, you choose to sit for the AS first. And then you sit for the 3 papers in AS, paper 1, paper 2, paper 3. Then you finish, in this case, you finish your AS level. Next June 2021, you will sit for the A2. In this case, you sit for paper 4 and paper 5. This is what's called modular. So you've done those AS and A2 in separate sessions. But some candidates choose to do this linear, like to sit for both AS and A2. In this case, you will sit for the 5 papers in one session. Now it's clear? Huh? Everything is clear
Yes,
now?
thank you. Thank you,
Huh?
Mrs. Hoda.
I can clarify again. I'm not annoying. I'm not annoyed, actually. Okay?
Everything is perfect. Yes, thank you.
Okay. Now, let me start now with our first topic, which is cell structure. You need a break or something, or should I complete? Is everybody okay with this?
Yes, please carry on.
Okay. Cell structure. We'll study the ultrastructure of the cell. We will know what are electron micrographs and diagrams showing the ultrastructure of the cell. In this chapter, as I said before, okay, we are going to know some facts about what are magnifications and resolutions. And as I told you before, I will teach you the detailed structures of the cell concerning cell surface membrane, nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, and ribosomes, and so on. Okay? First of all, I'm introducing the light microscope. This is what you are going to use in the lab, by the way. And I will talk about parts of it, not because they will ask you about in a practical exam. No, not at all. But because you need to know what is the eyepiece, what is the objective lens, and so on. This is what they call eyepiece. Sorry. Eyepiece. Here is it. Sometimes it can magnify 10 times or 16 times. This depends on the school. Okay, this part is called the objective lens. Okay, here is what's called the stage, and this is the place where we put the slide here, containing the object. On the objective lenses comes into actually 3 magnifications. You know this lens with the red line? This is what we call a scanning lens. And it can magnify 5 times. This one with the yellow line is the low power. And it can magnify 10 times. Another one which doesn't appear here in the image, this is the high power of your microscope. So take care when the questions ask you to make a high power magnification or a low power magnification, what to use. If the question is asking you to use the low power of your microscope, so this means that you are going to use this, low power. If he asks about detailed drawing, you are going to use the high power. Okay? When you use a light microscope to observe, for example, onion cells, this is what you see. What do you see now? You just see the cell wall, you just see the nucleus, and nothing else, I think. Do you see anything? Can anybody raise his hand or answer me? You can unmute now. What do you see in this image now? Susie, allow anyone to unmute to answer me if he has an answer.
Yes, it's done. They can unmute themselves.
Okay, who can answer me now? What do you see in this image? Not picking any. Ah,
Cell wall and nucleus.
just the nucleus. What's the color of the nucleus?
Blue.
Blue, yes. Do you see the cell wall?
Cell wall and nucleus.
Yeah. Do you see anything, any more details?
No organelles.
No organelles. You don't see any organelles, isn't it?
Yes.
Okay. Why is this? Why do you think is this?
It's a plant cell.
I know it's a plant cell, dear, because using a light microscope, I have what is so-called limited magnification and limited resolution. Again, light microscope is so-called because we use white light. In a light microscope, I have limited magnification sorry. Limited magnification and limited resolution. What is resolution? If I need to define resolution, it is the ability to distinguish between two objects seen as separate. Again, I will return to my major problem, is using the white light. I have limited wavelengths. Because you're using white light. You have just 7 spectra, and each with a certain wavelength, okay? And you may know that the lower or the smaller the wavelength, the higher the resolution. So again, if I am to use the light microscope, again, I have a very major problem. I have a limited magnification and limited resolution. This is why when I use the light microscope, I just saw a cell wall. I just saw the nucleus. I didn't see any other details. Unfortunately, we didn't see any other details. Okay? I return back to the next image. Okay, this is a plant cell. And also, this is an animal cell. You know what are those? Those are cheek cells. What do you see here? I need somebody to answer me. What do you see here in this specimen, please? Hi? Nobody's answering? Can I pick? Huh? I pick a participant to ask me? Sorry, okay, like I picked at random. Monawis? Mona, are you listening? Mustafa? Mustafa? Or Rahma? What do you see here, Rahma?
Hi, I'm Rahma. I see a nucleus, cell membrane. This is from the sheik cells, from the human sheik cell.
Yes, yes.
Maybe a cytoplasm.
Mm-hmm. What do you see in a
And
cytoplasm?
it's not quite clear, or maybe I don't
It's not
know.
clear. Good. It's not clear. You just see dots and that's it, isn't it?
Yes.
Okay, you see also, do you see the nucleus?
Yeah, the nucleus is quite clear.
Yes, and it's a blue patch, isn't
Yeah.
it?
Yeah.
Okay, do you see any details of a nucleus?
No.
No, just a blue circle, just a blue circle.
Yeah.
Okay. You know why we don't see details here? We are using light microscope.
Maybe because you're using light power, I get low power, I guess.
No, you know what? When I took this photo, I was using the high power. And in fact, yes, it was using, yeah, I took this photo using the high power. Okay? And even I can't see any more details. But what if I use a microscope with a higher magnification and a high resolution? Let's see together. Thank you, Rahma, for participating. Thank you.
Thank you, Miss. Bye.
Okay. What is this? This is the electron microscope. The electron microscope, as you see, is totally different from a light microscope. We use something called electron beam, which goes through magnetic lenses, which accelerate this electron beam. In this case, I can obtain a very short wavelength, and in this way, resolution increases. Okay, this is a diagram illustrate this, but actually this is the real electron microscope. The image here appear on a screen here or on this monitor. Okay, the sample usually is dead sample, and it's usually put here. And you know, we took a section through the electron microscope, and the electron beam passed through a vacuum. Okay, the sample is usually dead, and we see the images in black and white, but some attempts has been done to make false color like that, and we can see details. Another sort of microscope called a scanning electron microscope. This is a specimen for an animal or something, okay? And the electron beam goes in 3 dimensions around the specimen so that we can see the head of insect with these details, okay? See, we can see the hairs, the antenna, and you can see its eyes and its mouth parts and so on, okay? Could you see any internal structure here? Huh? Could you? Somebody answer. In this case, I can pick Sandra. Hi, Sandra.
Hi.
Hi. Do you see any internal structure, Sandra?
No.
No. You just see an outline of the insect head.
The external.
Just the hairs, just some parts of the eye and some mouth parts, and that's it. And the segments you see in the antenna, isn't it?
Yes.
You don't see it. This is why, because this image has been used, okay, sorry, taken using the scanning electron microscope. But if I need to see details, I need to use what's called transmission electron microscope. Okay?
Mm-hmm.
Transmission. This image of this electron micrograph, this is what we call electron micrograph. This image is taken using a transmission electron microscope. But the insect thing was taken using the scanning electron microscope. Okay? Now, can anybody tell me what you see here? If Sandra's still with me, can you answer me, Sandra? Are you still there?
Yes.
Hi, Sandra. Yes. Okay, now you can see nucleus, and now you can see some details, isn't it?
Yeah.
Yes. Where is Rahma? Is she still there?
Yes, Miss, I'm present.
Yes. Okay, now Rahma, I think you see more details in the cytoplasm, isn't it?
Yeah, it's
Okay.
quite more than the high-powered one.
High-powered light microscope.
Yeah.
Okay. And even while I'm using the electron microscope, I can go for each and every detail and I magnify it. You know why? Because I have unlimited magnification and I have a high resolution. You know what? Electron microscope has, okay, sorry. Has, oh, okay, what's happening? Sorry again. Has a resolution of 0.5 nanometer. Have you any idea about this nanometer unit? Rahma still there? Or Sandra, or I can pick another one? Can I pick another one? I can. Ahmed Ahmed, are you there? Camelia?
Yes, Miss.
Yes, Camelia. You know what's a nanometer unit?
It's a very small measuring unit.
Yes. You know what? We will talk about some units now. It's time to talk about them. Okay. 1 centimeter equals 10 millimeter. 1 millimeter equals 1,000 micrometer. You see how do I write micro? I'll write it again. Micrometer. And 1 micrometer equals 1,000 nanometer. Camelia told us now that nanometer is a very small unit, okay? And as I told you now that electron microscope has a resolution of 0.5 nanometer. You know what what does this mean? It means that if you have 2 objects and the distance between them is 0.5 nanometer, the electron microscope can make me see them as separate objects. But a light microscope has a resolution of 200 nanometer. Oh, you see the difference in resolution. Do you see this? Can I get a reply? Do you see the difference between the, a resolution in electron microscope and in a light microscope? Which one has less resolution now?
It's the light microscope.
Yes, because if the two objects has a distance less than 200 nanometer, a light microscope cannot make me see them as separate objects. This is why when we use the light microscope to see an animal cell, we just saw a nucleus, we just saw a cytoplasm which has granules, okay, which we saw just a cell membrane, and that's it. Isn't it? Okay, I'll return back to my show, to my PowerPoint thing. Okay? Is everything clear? Can I go more? Is there any question till now? Okay. Here's also a plant cell. You see here, this is the nucleus. You see how big it is. You see the chloroplasts. You see the vacuole. It's so big, okay? And if I take a look on what we call, this drawing is called artifact, by the way. Sometimes you see these drawings, you like them because of their color, but actually we didn't mean that, but it is for helping you understand the three-dimensional of structure of an animal cell. Here's photos like this are called false. Sorry, I'll take another color. False colored diagrams. You see these in textbooks, okay? This is to help you understand that the cell has 3 dimensions, okay? You see, this is a beautiful image of a nucleus, and the tiny ball inside is called nucleolus. This part is mitochondria. And here is a smooth endoplasmic reticulum and rough endoplasmic reticulum and so on. I have the same image for a plant cell. Clear the drawing, okay? This part, okay, this part is the large central vacuole. And here is the chloroplasts. If you try to take the same image under a light microscope, you will not see these things inside chloroplast and even in the mitochondria, okay? And here appears the rough endoplasmic reticulum, and here's what we call Golgi apparatus. Let me go in more details, if you don't mind. This is the plant cell, and let me go. Yes. Do you see the difference between an animal cell and a plant cell? Who can tell me the difference, please? Who can tell me? Can I pick at random? I have here Farouk. Are you there? Farouk Ibrahim? It's not there. Are you there, Farouk?
I'm there, miss.
Yes. Do you see the difference between an animal cell and a plant cell? Just give me one difference.
Animal cell have only cell membrane and don't have cell wall, while plant cells have cell membrane and cell wall.
Okay, what else do you see in the diagram? Give you a difference between animal cell and plant cell.
So, 1 second, please. Plant cells have chloroplast, while animal cell have no chloroplast.
Yes. Now, okay, Farouk told us that there is a chloroplast here and there is a cell wall here. Is it, Farouk? And did you see the vacuole?
It is not, the vacuole is present over here.
Yes, it is. Here it is, isn't it?
Right.
Okay. And there are a lot of common things between animal and plant cell. Could you tell me something which is common in between? Huh?
Common.
Common, yeah? Yeah.
The two cells have cytoplasm, the two have nucleus,
Yeah.
the two have Golgi apparatus,
Yes.
the two have vacuoles and mitochondrion.
That's good. Thank you, Farouk. Okay. I will return again for these differences, okay? But let me go for another part. You see, I told you before, when I use a light microscope, and I think you are familiar with this section, this is section in a leaf. You see, these are the palisade cells. Do you see any details within the palisade cells other than I have just green patches? I don't think so. And even if I try to scan it and watch more and more, I'll just see some green parts, and that's it. Okay, but let me go for another image. Oh, what is this? It's a scanning electron micrograph. I use the scanning electron microscope. And now I can see nucleus with some holes on it. And I can see some patches concerning the chloroplast, and the empty thing is the vacuole. Can I see internal structure? I don't know. I don't think so. I cannot see the internal structure of a nucleus because I'm using the scanning electron microscope. But if I use the transmission electron microscope, I will see more. Oh, now I'm using a transmission electron microscope, and I'm using a high magnification so that it helps me to know details of a nucleus. Watch with me. I am now going around the nucleus. Here is what is so-called, is it clear for you? No, I don't think this color is okay. Let me use the orange thing. Okay, here it is. You see? All of this I'm going on is the nucleus. Do you remember, Rahma, when we used the light microscope, we saw just a small patch within the cell, isn't it?
This
Rahma,
one
are you
looks,
there?
yeah, this one looks like the cell. And I can't even imagine that this is the nucleus I saw from before. How cool.
Yes, yes. You know what, Rahma? This is because you are using now a transmission electron microscope with a high magnification and high resolution. And now you see it large as this, okay? Do you
Yeah.
see this part, which is called nucleolus? Okay.
This is the internal part of the nucleus.
Yes, yes. You see what? The nucleus has a baby now, which is a nucleolus. Okay. No, it's not a baby. I'm just calling it a baby, but actually it's not. It's a separate structure. Okay.
Inside the nucleus.
Yes, it's like it, but it's not.
Yeah.
Okay, it's part of it. But now you discover that the nucleus, okay, has dark areas and white areas. What are those? Let's see together.
Okay.
Now here it is, the nucleus, and it is surrounded by what's so-called nuclear envelope. You know why? Because the nucleus has two membranes. It's a double membranous organelle, okay? The white areas, we call it euchromatin, and the dark areas are called heterochromatin. And as I told you now, this dark patch is called nucleolus. What is a euchromatin and what's a heterochromatin? Okay. All we know from our previous study that a nucleus contains the DNA and contains the chromosomes, isn't it? Okay. So, do you think a nucleus in a cheek cell does the same function as a nucleus in a pancreatic cell? Who can tell
No.
me? Huh?
No, it has different code, I guess. For يعني it
Good,
codes
good.
for a different function.
Yes. So, you know that in pancreas, the pancreas sometimes parts of the cells works to produce insulin or glucagon. They produce hormones, and other parts in the pancreas produce digestive enzymes. And your cheek cells have a totally different function. They produce mucus. Okay? So, do you think the DNA works the same way in all these types of cells? Of course not. So how this difference in function happens, this is what we call differentiation. And euchromatin is the areas where DNA is active, and heterochromatins are the areas where DNA is inactive. Look at the other nucleus in the next cell. This is the one on the left side of the, right side of the screen, okay? This one, for example. At this one, at this nucleus, okay? Does it have the same distribution of euchromatin and heterochromatin? Do you think?
Absolutely not.
Of course. Now, can I can know that this cell does a different function than from this cell, because distribution of euchromatin part is not the same as this parts, isn't it? So when you take a sample from a cheek cell or a pancreatic cell and you observe them under electron microscope, you see a different distribution of euchromatin and heterochromatin. Okay, all of us, we know that we came from one cell, which is the zygote. But after the zygote has developed into an embryo and then a fetus and so on, some cells made some tissues and organs of the nervous system, of circulatory, of digestive, and so on. How this happened? This happened when DNA, okay, some of it were distributed in the euchromatin areas and some are distributed in the heterochromatin areas. So again, the white areas are the euchromatin, okay? This is where DNA is active. Heterochromatin, dark patches, this is where DNA is inactive. And what about the nucleolus? Is it only the baby of a nucleus? No, it has a separate function, which is formation of an organelle called ribosome. Have you ever heard about ribosomes before?
Yes, it's for making protein.
Yes, yes, in protein synthesis. Okay. So nucleolus has its own function, its separate functions away from the nucleus, okay, which is formation of ribosomes. Okay, so now it's not the baby of the nucleus, huh? Okay. Let me go for the next slide. Again, I picked the another image from a different cell. You see now there is, well, it's very obvious that this cell is metabolically active. How did I know? Farouk, where are you?
I'm here, Miss.
Okay. I'm saying now that this nucleus is taken from a cell which is metabolically active. How did I know?
Main number of ribosomes and the black regions.
No, I think you meant the white regions. This is where DNA is active. I don't have ribosomes here, by the way.
I am
Okay, I'm figuring out. Oh, yeah, I know. So euchromatin areas, you see the white patches here? Okay, this is where DNA is active. So this cell is all the day, the DNA is coding and protein synthesis is going on and so on. Okay, and you have very few heterochromatin areas. Okay, this image, I got it from a scanning electron microscope. In this image, okay, a beautiful thing here is obvious, which is the nuclear envelope. And we discovered at this moment that nuclear envelope is not continuous. It has pores in it. Do you see these pores? Do you see those pores? Okay, we call it nuclear pores. Okay, don't say holes, it's different. Okay, now, so this image, I mean, it is taken by scanning electron microscope. Okay, do you see details here, Farouk? Or Sandra, or Rahma? Huh? Do you see any details?
No.
Do you see the hetero and euchromatin here?
No.
Do you see the nucleolus?
No.
Okay, you know why? Because I get this image using scanning electron microscope.
Microscope.
I just see the nuclear envelope with the nuclear pores. Why it is so porous? Because Farouk told us from a moment now that ribosomes are made in the nucleolus, which are necessary for protein synthesis. You know what? When ribosomes are formed, parts of it, they go out of the nucleus through these pores. Through these pores. I'm coloring it. Okay? Also, during protein synthesis, there is a molecule made in DNA, so in the nucleus, called the messenger RNA. It comes out through these pores. Okay, this is why the nuclear envelope is porous. This we call them, as I said, nuclear pores. Is everything about the nucleus clear? And this is also another scanning electron micrograph, but actually it's false colored. Okay, everything about the nucleus is clear. I hope so. Is there any question?
No
Susie?
questions, Mrs. Hoda. Thank you.
No questions? Okay. Now I returned back to transmission electron micrographs, and I saw that the nuclear envelope has nuclear pores. Okay. As I told you before, this is to help the parts of ribosome to go out through these pores so that they stay in the cytoplasm, as well as the messenger RNA. Okay. This is a higher magnification so that I can see the nuclear pore in details. Out in the cytoplasm, I'm going for another organelle, which each and every cell needed, which is the mitochondria. And yes? Okay, did you get enough today? Should I complete? Susie, is there any objection to complete?
No, nobody is complaining.
Okay, fine, I will complete. Okay, we'll see these are the mitochondria. Okay, and of course I got this image from a very actively respiring cells because mitochondria are for aerobic respiration. This is where ATP is produced. And later on, I will refer to what is ATP. Okay, let me increase the magnification. And take an, okay, it's a diagram, illustrates the mitochondria from inside. And I will see that it has two membranes, inner one, which is folded, and outer one, which is smooth. And I will go further and further within a mitochondria, and I will take a 3D image for mitochondria. I will see that the inner membrane has folds, as you see, like finger-like projection, and the outer membrane is smooth. What is this? Okay. The outer membrane, which is smooth, and the inner one is folded into structures called cristae. Okay, cristae is plural, but crista is the single. So again, this one and this one are called cristae. Cristae for plural. Sorry. Single is crista. Why do I see these folds? I told you now that the mitochondria is the site for aerobic respiration. Okay. Inside here, there is no space. There is what's so-called matrix. Aerobic respiration goes through many steps. First step happens in the cytoplasm, and there are 3 steps there inside the mitochondria. The folds inside and the crista, this is to increase the presence of crista. What's going on? Yeah. Okay, the presence of these crista, okay, is to increase surface area for enzymatic reactions. The more crista or cristae the mitochondria has, the higher the rate of enzymatic reactions. And you need this. These reactions are called oxidative phosphorylation. We need this because by this enzymatic reaction, I will produce more ATP. So guess what? If I take the image from a mitochondria in a muscle cell, I will find a lot of cristae more than I take an image from a mitochondria in a skin cell. Why? Muscles all the day do the contractions, help you to move your organs and so on. So they need a lot of ATP to do this contraction thing. But your skin cell, yes, it's respiring and so on, but it doesn't need this so much ATP. Okay? Is this clear? In the matrix, you find what? You find a fluid. In this fluid, you will find a circular loop of DNA, and you will find ribosomes. What is this? Is there a DNA and a ribosome inside the mitochondria? Yes. Let me illustrate something. sorry. Okay. You know what? So usually, sorry. Sorry, I'll get another one. Tell us something about the cell structure. Cell structure shows compartmentation and division of labor. You know what this means? Compartmentation, that you have membranes separate, sorry, separate organelle components from cytoplasm. And this was very obvious talking about the nucleus and mitochondria. Division of labor means that each organelle has its own separate function. Okay? Again, for the mitochondria, as I said before, it has an outer smooth membrane and an inner folded one. Mitochondria, as I told you before, these folds, each and every one is called crista, and plural for cristae. Okay? I told you that there are many cristae here. Why? This is to increase rate of enzymatic reactions. Okay. Enzymes are proteins in nature, isn't it? And they need to be synthesized. Do you think for a mitochondria to do its function, it's gonna wait for a protein to be synthesized from out in the cytoplasm, or it's gonna do it by itself? Sandra, or Mona, or Farouk, or Rahma? I'm sorry I didn't pick all the names. Huh? Where are you?
Do it by itself?
Yes. So this is why inside the matrix you have a DNA and its own ribosomes. This is why to make its own proteins. Okay, this really helps, okay, the mitochondria to do its function. Is this clear? Huh? Is this clear?
لا دي فري سيشن احتمال كبير تلاقيها او ليفل مش اي ليفل مش عارفه
Hi. Hi. Need to mute. Huh? Is this clear? Hi, May. Okay. I'll go for the next slide, and you will see again an electron micrograph of a mitochondria. Okay. As I said, I have outer smooth membrane. I have an inner one, which is folded, okay? And I have here the matrix, which contains DNA and ribosome for the mitochondria to make its own proteins. The folds, or the more folds I have here, okay, the more the ATP is produced by mitochondria. You see those extensions in the crystal? You see those spots? This is what we call the stoked particles. This is where the enzymes are there for the reactions that happen on the inner membrane of mitochondria. Again, I return it to a photomicrograph taken by a light microscope, and I am seeing some plant cells, and I see just green circles here. You know what's those? These are the chloroplasts. Do you see any more details? Of course not. This is why I need to use an electron microscope, yes, to find out the details about a chloroplast. Here again, I have a double membranous structure as an organelle, which has a double membrane, an outer smooth one and an inner one with folds, okay? But here the folds are stoked over each other. Each layer is called a thylakoid, and the stack of thylakoids, the stack of thylakoids here is called granum. This diagram has meant to make the granum green. You know why? Because on these chloroplasts, there are the photosynthetic pigment, which is called chlorophyll. This is the place where light-dependent reaction happen, and because chloroplasts are the site of photosynthesis. Okay, in electron micrograph, the structure of chloroplasts is totally different because, you know, this previous one, just as I told you before, a false color diagram to show the inside of a chloroplast. I'll stop at this structure today, okay? And if we are lucky to meet again in a new lesson, I will complete this chapter and explaining more and more organelles. In the next lessons, we will see more details about chloroplasts and how they are adapted to their function. And this is a scanning electron micrograph, so inside of a chloroplast. This one is false colored. And I will also talk about the rough endoplasmic reticulum and its function, how it looks under electron microscope. And this is a three-dimensional image of it, okay? And what's the function of a rough endoplasmic reticulum, as well as the function of a smooth endoplasmic reticulum. Then we will go to see more details about a ribosome and its function and the structure of it. This is an image of many ribosomes together. And we'll go through a detailed structure of the Golgi apparatus, and we will know the detailed structure of a Golgi apparatus and how it works. And then we'll know a new structure called lysosomes and details of a cell membrane and the cell wall. And in this way, we will end the cell structure. Okay, I hope you enjoyed the lesson for today. And if you have questions,
Yes, thank you,
I'll be
ma'am.
happy to answer. I have some questions to do together. Are you ready for those? Are you ready for those? I hope so. Okay. I needed to show you how the question goes in paper 2. You see, these are what we call electron micrographs of some organelles. What is this organelle? Could you write your answer?
Yeah. Mitochondria?
This is the mitochondria. What's the function of it?
Aerobic respiration.
Okay, so if I need to state the function of it, it's aerobic respiration or production and synthesis of ATP. As I said before, aerobic respiration is a huge reaction. It starts in the cytoplasm and ends in mitochondria. So that to be precise in our answer, okay, I don't say that you have a wrong answer, but you need to make it more precise. As I said before, aerobic respiration are 4 stages. First stage starts in the cytoplasm. It's a huge process. You're going to study it in A2, okay? But here, if you need to precisely mention the function of a mitochondria is production, or you can mention synthesis of ATP. Is this clear? Okay. About this organelle C, what is this? Huh? What is this? I need an answer.
Muscle cell?
No, I mentioned an organelle. This is an organelle. This is the last organelle I spoke about. Chloroplasts. Synthesis and production of glucose or the site of photosynthesis reaction. Okay, look at this question. Figure 1.1 is labeled diagram of a leaf palisade mesophyll cell as seen with a high quality light microscope. An electron micrograph of the same leaf mesophyll cell at the same magnification would show more details as shown in figure 1.1. Why at same magnification, electron microscope is able to provide more detail than a light microscope? Do you remember?
Because it has resolution?
Yes, good. This is because of resolution, yes. So my main hit here for the examiner is referring to resolution. I can mention here in electron microscope. Do you remember the figure I told you? Huh? Has a resolution of 0.5 nanometer, but light microscope has a resolution of what? Huh? Do you remember the figure?
200?
Yes, 200 nanometers. So I can refer that this one has a low resolution compared with what? Huh? With electron microscope. Okay? Let me go for the next part. Describe 3 additional features that can be seen with on electron microscope of the mesophyll cell. What can I see? As I told you before, I can see more organelles in the electron microscope. Could you mention 3 organelles I can see using electron microscope? Huh?
Chlorophyll?
Chloroplast. Details of chloroplast, yes. What else? Huh?
Details of mitochondria.
Yes, good girl. Details of mitochondria. What else?
Details of the nucleus.
Yes, excellent. Thank you. Can I go for another question? Who can name me organelle labeled B?
Nucleus?
No.
Nucleotide.
No. Huh? Rahma, you remember the baby of the nucleus? What was so called? Huh?
Nucleolus?
Nucleolus.
Yeah.
Yes. You know the plural of nucleolus? Nucleoli. I need to mention something. Take care of the spelling of organelles. Nucleolus, nucleolus. Sometimes you lose a mark because of misspelling. Silly marks. You know what? I hate silly marks. Okay? When you lose a mark, you lose it because it was a hard question. But please don't lose a silly mark because you messed up the spelling. Okay? What's the function of a nucleolus?
Ribosomes production.
Yes, good girl. Okay, can anybody tell me what is organelle C?
Mitochondria.
Yes, mitochondria. Again, what is the function of a mitochondria? Huh?
Producing ATP.
Good girl, that's it. This is, okay. You know what I like about this chapter, cell structure? That the questions are so direct. They're not that type of questions that you think real hard about this. I know we didn't go through the other organelles, okay? I will postpone the rest of the questions to another lesson, okay? I hope to see you in another lesson, God willing, inshallah, okay? Thank you.
Thank you so much, Mrs. Hoda, for this incredible session. Inshallah, if anyone wants to register for your course, they can follow our Facebook page or go and visit our website. I posted the website on the chat, so they can just click on it and then they can register for your course. Thank you so much, and thank you for everyone who participated in this session.
I see Sandra raised the hand. What's Sandra? Excuse me, Susie. Sandra raised the hand. Yes, Sandra, how may I help you? Sandra? Not here, okay. Is there any more questions?
That's it, Mrs. Hoda. Thank you so much.
Oh, yes, I want to ask a question.
Yes.
Can you send the website again in chat?
Send what?
Yeah, sure. The website.
The
I
website.
will resend it. Yes, of course.
Okay.
Thank you.
You're welcome.
Any more questions? You need to know something before I leave, that I will make separate lessons for practicals, okay? But this is concerned the chapter. When I finish a chapter, okay, I will make a whole lesson for the practicals and the ideas and how do you manage practicals concerning this chapter, okay? So that we can cover the 3 papers of AS, okay? Thank you. I'm done, Susie. If anyone has more questions, I'm done. If you have any questions.
They are just asking about OL, Mrs. Hoda.
O levels, yes.
Yeah.
When is the free lesson?
Yes, please. Is going to be one? Are you planning for a free session for OL biology?
Yes, I am. Okay. I think we will. I will tell you, and you will post this
on our Facebook page.
Yes.
Okay. So there will be an OL session, free session, and the date and time will be posted on our Facebook page and on our WhatsApp group as well.
Okay. Inshallah, tomorrow at the same time we will have another session, free lesson for A2. And we will. I will tell Susie when is the free lesson for O level.
Okay. Thank you so much, Mrs. Hoda.
You're welcome, Susie. Thank you.